The human nervous system processes an overwhelming volume of simultaneous inputs and outputs. To prevent signaling pathways from colliding and causing physiological chaos, the central nervous system operates on a rigid, highly segregated anatomical blueprint. At the core of this blueprint is the spinal cord, which functions as a strict one-way highway system. Looking at this somatic pathway diagram, we trace the directional flow. Afferent, or incoming sensory signals, exclusively enter through the back, the dorsal side, of the spinal cord. Efferent, or outgoing motor signals, take a different route, exiting strictly through the front, the ventral side. This segregated directional flow feeds two distinct tiers of processing. Localized spinal loops handle immediate reflex responses for speed, while ascending and descending tracts connect to the cerebral cortex for conscious voluntary control. Mastering this exact geographic segregation, dorsal for input, ventral for output, is the inviolable foundation of clinical neuroanatomy. If a specific nerve pathway is damaged, the physical location of the resulting deficit tells a clinician exactly where to look in the spine. Every conscious physical movement you make requires spatial awareness. Before the brain can issue a motor command, sensory data must translate the physical properties of the outside world into an electrical stimulus the central nervous system can read. Focusing on the top left of our diagram, this afferent chain begins with highly specialized peripheral receptors. Muscle spindles directly detect mechanical stretch, Golgi tendon organs measure tension levels, and free nerve endings translate pain and temperature. The tactile feedback of the skin is mapped out just below that. Meissner corpuscles process light touch, Pacinian corpuscles capture vibration, and Merkel cells register steady pressure. Once the physical stimulus triggers an electrical signal, it travels along the afferent first-order neuron. The cell body of this specific neuron sits completely outside the spinal cord, gathered in a cluster called the dorsal root ganglion. From the ganglion, the axon plunges into the central nervous system, strictly passing through the dorsal root and terminating in the dorsal horn. These afferent signals do not enter the spine randomly. They arrive at specific vertical segments corresponding to body geography. Receptors in the arms wire directly into the cervical spine, the legs route into the lumbar levels, and the pelvis connects to the sacral segments. Because this wiring architecture is absolute, a patient's inability to feel a specific sensation in a specific limb allows clinicians to pinpoint the exact vertical level and receptor type failing in the afferent chain. If you brush against an open flame, routing a pain signal all the way up the spinal cord to the brain, waiting for the cortex to formulate a decision, and sending a motor command back down would take hundreds of milliseconds. By the time you snap your hand back, severe cellular damage has already occurred. To prevent this, the nervous system uses reflex arcs. These pathways completely bypass the conscious cortex. In a reflex, the spinal cord acts as a localized supercomputer, executing the necessary motor response on its own. The cortex finds out about the threat milliseconds later, well after the limb is already safe. The central panel of our diagram maps out how this localized processing works, starting with the fastest circuit in the human body, the patellar stretch reflex. A tap on the patellar tendon stretches the muscle spindle. The afferent sensory neuron fires, enters the dorsal horn, and synapses directly onto an alpha lower motor neuron in the ventral horn. Because there are no middlemen between the sensory input and the motor output, this is a monosynaptic circuit. Handling the open flame requires a slightly different mechanism, mapped below as the withdrawal reflex. This circuit is triggered by pain or sharp trauma. The withdrawal reflex is polysynaptic. The afferent pain signal enters the spinal cord, but instead of connecting directly to the motor neuron, it first synapses onto an interneuron located in the intermediate gray matter. This single interneuron performs two critical actions at the exact same time through a process called reciprocal inhibition. It simultaneously activates the flexor motor neuron to quickly contract the limb and pull it away, while actively inhibiting the motor neuron of the opposing extensor muscle. If reciprocal inhibition did not exist, the massive electrical surge from the pain stimulus would cause both antagonist muscles to contract simultaneously. Your arm would lock rigidly straight into the fire. Suppressing the extensor guarantees a rapid, smooth withdrawal. Reflexes exist for immediate, unconscious survival, but navigating the environment requires conscious, voluntary motor control, and that requires the efferent pathway. The right-hand column of our diagram maps this descending route. Every voluntary movement originates deep inside the frontal lobe of the cerebral cortex, specifically within the primary motor cortex of the precentral gyrus. The neuron firing here is the upper motor neuron. It drives the command downward using the excitatory neurotransmitter glutamate, while local GABA networks provide necessary cortical inhibition and modulation to refine the movement. The electrical command leaves the cortex, traveling down through a wide fan of white matter called the corona radiata, before funneling into a tightly packed architectural corridor known as the internal capsule. Because so many descending motor fibers are compressed into this tiny anatomical bottleneck, the internal capsule is a notoriously high-yield hotspot for strokes. Even a highly localized lesion here severs communication to huge portions of the body. Fibers that survive the internal capsule continue their descent through the midbrain and pons, arriving at the medullary pyramids at the base of the brainstem. Here, the most critical navigational shift in the nervous system occurs, pyramidal decussation. At this exact junction in the caudal medulla, the vast majority of descending motor fibers cross over the midline to the opposite side of the body. After decussation, the signal descends down the spinal cord via the lateral corticospinal tract. Upon reaching its target segment, it enters the ventral horn and synapses directly onto the lower motor neuron. Because of this exact crossover point at the medulla, neurological deficits follow a strict rule. A stroke localized in the right hemisphere of the brain will always manifest as motor paralysis on the left side of the body. Reaching the lower motor neuron introduces the defining rule of somatic motor control. The central nervous system uses exactly one neuron to bridge the gap from the spinal cord directly to the target skeletal muscle. Where this single neuron meets the muscle tissue is the neuromuscular junction. As the electrical action potential arrives at the motor axon terminal, the sudden shift in voltage forces calcium channels in the membrane to snap open. Calcium floods into the terminal. This influx forces internal storage vesicles to migrate to the boundary and dump the neurotransmitter acetylcholine directly into the synaptic cleft. The acetylcholine diffuses across the microscopic gap and binds to nicotinic N-M receptors, which are anchored directly on the muscle's motor end plate. Because these receptors are ligand-gated channels, the physical binding of acetylcholine forces them to unlock. Sodium ions rush into the muscle fiber, creating a localized depolarization known as an end plate potential. If that end plate potential hits threshold, a new muscle action potential fires. This signal commands the sarcoplasmic reticulum to release its internal calcium stores, sparking the actin and myosin cross-bridge cycle that physically contracts the muscle fibers. This exact mechanism is highly restricted. The somatic pathway strictly targets skeletal muscle, the specific tissues governing your quadriceps, your facial expressions, and your diaphragm. This single neuron, acetylcholine-driven chemical bridge is the absolute only mechanism the conscious brain has to exert physical force on the outside world. The architectural elegance of this somatic pathway becomes obvious when you compare it to the rest of the nervous system, specifically the autonomic nervous system. While somatic output relies on a one-neuron direct line to skeletal muscle, autonomic output always requires a two-neuron relay. In the autonomic relay, a preganglionic neuron exits the central nervous system but stops halfway at a junction called a ganglion. There, it releases acetylcholine onto nicotinic N-N receptors. From the ganglion, a second postganglionic neuron travels to the organ. If the body is triggering a parasympathetic resting state, this second neuron also uses acetylcholine, but targets muscarinic receptors to slow the heart or stimulate digestion. If the body needs a sympathetic fight-or-flight response, the postganglionic neuron switches chemicals completely, releasing norepinephrine onto alpha and beta receptors to dilate the pupils and accelerate heart rate. The target tissue dictates the architecture. The complex two-neuron autonomic system automatically drives smooth muscle, cardiac tissue, and glands. The simple one-neuron somatic system exclusively drives voluntary skeletal muscle. The nervous system perfectly segregates voluntary physical force from involuntary physiological life support by enforcing strict anatomical routes. From the dorsal entry of sensory data to the localized processing of spinal reflex loops, down to the exact neuron count required to move a muscle, the architecture ensures the signal always hits its mark.